This is Dr. Ben Morrill. Welcome to episode 21 of Reptile Genetics Weekly. Glad to have you here. Please, if you enjoy this, like and subscribe. And for those DNA nerds out there that are already subscribed, thank you so much for joining us. And today we are going to do something that I think a lot of people will find interesting. At the Pomona show, I broke out my laptop and showed a few people real briefly how it is that I analyze the data when I'm doing morph tests, so people could kind of get an idea, what this looks like and what does a normal versus a het versus a homozygous look like. So I thought that at some point I want to do that for you all. And since we finally, I haven't been out of town or sick, I could kind of do some prep for tonight. So we got this prepared for you. But we do want to do a weekly update for results. So those of you that gave me sheds at Pomona, I do have results. I haven't sent them out to you yet, but I'll try to do that tonight before I go to bed. Make sure you get your results from the sheds that got to me at Pomona. We did do a research run this week, so it was only the few sheds that I got at Pomona and that I had, just kind of odds and ends that I had here. But the majority of it was doing research sequencing for new morph tests. And it was a good run. And hopefully next week we can talk about the results from that. It might take a little longer, but very soon we'll have some new tests available finally. And over the next month to 6 weeks, we should have several more. We'll have a bunch flood in as long as things keep going well like they have. So, but the next run will start mid to late next week, this week, I mean. So by the end of this week, we'll have another run going. And so probably about a week from today, Monday or Tuesday next week, will be the next batch of results for morph testing. And for sex determination for colubrids and ball pythons, I know that some of you have been waiting a while on that. We are kind of working on how to do that faster and more efficiently, and add another person that has the skills to do that so we can do it. I'm hoping in the next 2 or 3 weeks, maybe a month, we can get to where sex determination results will always be 2 weeks or less. So we should be able to get to where we're running them once a week. And so if your sheds happen to come in the day after we do a run, the longest wait you should have would be waiting a week until we're running again, then a couple of days for us to analyze. So it is because we're improving the sex determination testing to make that faster, but it has been slow. But I will work on those tomorrow to try to get everybody caught up if you're missing sex determination results. So it's part of having a small startup business like this and growing it fast and have a small number of people. We're very thankful to all of you that have been patient with us. We're, first of all, super excited that the tests we thought would be helpful are helpful. And we definitely think about you every day when we aren't able to turn results around fast, but we are working on making that faster. So we're chipping away at it every day. And so, yeah, the sex determination results, I'll hopefully have those for everybody. We're waiting on those. Now, if you just sent something in for sex determination in the last week, that probably isn't to me yet. And so that'll be, another week and a half until you get those results. But anyone that's been waiting over a couple of weeks, you'll get those results finally. So yeah, then for today's episode, I actually have not only Kayla, but I have Dr. Travis Wyman here too.
Hi, Ben. Hey, everybody.
So the reason we invited Travis to come on here is because we are going to do a little bit of a demonstration like I brought up earlier, like I did at the Pomona show, be able to show you all what it looks like. I'm sure many of you have thought about and wondered, what does this genetic test look like? What is it that Ben is actually looking at when he's testing my snakes? And can he really take a little tiny piece of shed that's only a couple of scales and get a bunch of genetic information from it? Well, we'll show you tonight what that looks like. And Dr. Wyman has got lots of experience working with next generation sequencing and alignments and analysis. And I think that he is the kind of person that if he told you everything he did, he'd have to kill you. So he can't tell you everything.
Neither confirm nor deny.
He does some pretty cool DNA stuff, and I'm sure he'll have some good information to add as we kind of go through this. So the gold standard and what's been done for decades is Sanger sequencing. And what was done before that was even more painful, but we'll start with Sanger. And so with Sanger sequencing, there's 4 different bases that we're looking at: A, T, C, and G. And when you do a Sanger sequencing run, you get for each sample, you get one reaction that gives you one read. You're essentially having a bunch of different fragments that are made, and each one where it terminates, you get a color. And so on the screen here, you see these different colors. Red is a T, blue is a C, A is green, G is black. So you have different colors. You have the bases there above. So this is what Sanger sequencing data looks like. And if you look on the top line, in this, we don't use Sanger sequencing. So I'm taking this as an example online. And so this is, the top line is a normal. The middle one is a het. So you can see the top line just has a T, has a nice big clean T peak. So for that specific base, which in this case is 821. So base number 821 is a T in the normals. And then in the heterozygotes, the middle line, it's a T or a C. So that's why you have a lower peak there, and there's actually 2 peaks. There's a T and a C. So that's your heterozygote. And then the bottom one is the homozygous mutant, whatever it is they're studying. So everything is a C. So you get one nice big blue peak. And so this, like I said, this is the way we've done sequencing for decades. And over the last, what would you say, Travis, 15 years, 20 years? It hasn't been 20 yet, has it?
Well, I guess it depends on how far back you go with the next gen type of stuff. Early 454 stuff, we probably could call it 20 years.
Yeah.
But definitely the last 15 solidly.
Yes. So about the last 15 to 20 years, we have what Travis brought up, next gen sequencing, next generation sequencing. And there's multiple platforms that do it. The one that I've used the most is Illumina. I don't know. What is it that you all run?
We primarily run Illumina, but we also run PacBio and a little bit of Oxford Nanopore.
Okay. So yeah, there's multiple platforms, and each one kind of has some pros and cons. Illumina is the one that I've used for the last about 15 years. Definitely the last 10, I've been using it in the full-time job I had at a pharmaceutical company. So when we are looking at next-gen data, it's a lot different than what we just saw. And instead of having one sequence where you have fragments that end, all along the way, and you get one color coming off for each base, we're able to get thousands to millions in the really big runs of reads per sample. And you can see each DNA molecule individually. And I'll show you that in a second, as long as you all will take my disclaimer right now and warning not to get a headache when you see it. Don't be stressed. I'll click a button that'll make it look cleaner and not as scary. But yeah, it gives you a ton of information, even from just a couple of scales. Once we extract that DNA and sequence it with this powerful technology, we can get a ton of information, and you can see very clearly whether something is het or not. It's not something that you're going to easily mistake if you have a good sample and you get good results back. So what I'm going to do, I'm going to share my screen. So it's going to kind of do the screen within a screen within a screen for a second here. And then I will show some DNA information.
Yeah. For those of you who aren't familiar with this sort of thing, neither am I. It's the first time I've seen it. So we're in this together, guys.
Okay. So is it showing?
Yes. There we go.
All right. So here on this top line where it says pi WT, that's for wild type. So this is the TFEC gene in the region where the mutation is that the group, Dr. Heather Rafi and Dr. Alan Garcia Elfring, they are the ones in Canada that found the PIDE mutation. It's been a few years now. since they published that.
And that's the same gene we talked about in our second episode of RGW, I think.
Yes.
Or when we first started doing this in piebald, anoles, and was it mice as well?
Mice, yeah.
Yep.
Yeah. Okay.
Yeah, so this is just about 300 base pairs. As you can see here, it's about 300 base pairs of sequence. On this top line, that's what a normal looks like. On the bottom line, this is a pied sequence. And if I just scroll through it like this, you're like, well, everything looks, sorry, you guys get to see my mail. So you can see everything looks the same, but if we put these dots in, what it does is it puts a dot wherever something is identical, and then we can see at base 141 in a pied animal, there's a T there instead of a C. So that's really, out of all the stuff we're doing for a pied sample, we're just looking for whether there's a C that changed to a T, and whether we see just T or we see C and T, or we just see a C. So those are the three things. If we just see a C, that's a normal. If we just see a T, that's a visual pied. If we see both, that's a het. And so I will show you some examples. So in this one, so this is an actual animal. This is sample number 2 on this run.
It's kind of scary looking.
Yeah, so this is the part I told you not to get too worried about yet. So if I don't have the dots on, it just looks like a whole bunch of stuff. With the dots on, it helps us see, what's potentially different. So on each run, you can see where my scroll bar is moving. As I move through this is like 5,000 reads. So for this one sample from a couple of scales where I extracted the DNA, I'm getting thousands of reads. And in this case, like we saw on the last screen, 141 is the one that we're interested in, right? And so in this case, everything is a C. So it's very clean. This animal is a normal. So if we go to the next example, so this is animal number 3, and we get past these kind of garbagey reads up here to the good ones. Once again, we've got thousands of reads here. And what do we see? We see all T's at base number 141. So this is an example of a visual pied. So that's an actual pied.
Cool.
So it looks very different from what the normal looked like, and it's very clear. And I have like 5,000 independent things saying this is a pied. It's very obvious. And in this last one, get past the garbage reads.
So what causes the garbage reads? You said that was like is it a damaged sample or uncertainty, or what's?
I mean, this is still a great sample, because if you look at my scroll bar, the garbage reads, there's like maybe, I don't know, 300 of them, something like that, 200 or 300 garbage reads.
Several thousand good ones.
Yeah, and then there's still like 4,500 that are all clean. There's going to be little mess ups here and there in some of the reads, but you have so many to look at that it's obvious what the sequence is.
Yeah.
And when we look at 141, we have about half of them are T's and about half of them are C's. So this animal is a het pied. And unlike the Sanger, where you have those, peaks that you're looking at, here you have hundreds to thousands of independent DNA molecules that are telling you what your answer is. And so it's very, very clear. It's extremely unlikely. I'll go ahead and hide my screen now. Or let's see. It didn't switch back, did it?
No.
There we go. Stop sharing.
There you go.
All right. That's why I have Kayla run this. I'm not good at it.
That's okay. You did fine.
So yeah. So it's very, very clear. And you have lots of information to go off of. And it's extremely unlikely that you're going to have problems. And as we've talked about on other times, really the mistakes come in people either when they're sending the shed in, or when we're preparing the shed, or when we're reporting the results, something gets mixed up. It's extremely unlikely and rare that the test itself has a problem, because it's so obvious what the answer is.
Yeah.
So yeah,
that's cool. So you're basically staring at those base pairs all day when you're doing this analysis, right?
Yeah, it's sometimes. The other day, my wife, I think it was like, I don't know, 2 or something. I'd been up since 6 or 7 in the morning analyzing, and she walked in, and I had my feet up on the ottoman, sitting laid back on the couch. I'm wide awake, but just like physically exhausted just from staring at DNA sequence for, I guess that would have
You
been
start to dream in A's, T's, G's, and C's.
for sure.
Sure.
You have like little nightmares of just screens full of A's, T's, C's, and G's. Wake up with the sweats that you're surrounded by A's, T's, C's, and G's.
The life of a geneticist.
Yeah. It is cool because of all of the stuff we can figure out with it. But yeah, it's sometimes if you do it long enough in one sitting, it can fry your brain.
Yeah, little bit.
So just out of curiosity, so the benefit of the next gen sequencing is that if you were to run a panel, you could see multiple lines where like you could see a bunch of T's and then a bunch of A's, wherever that mutation is. And that's
the nice thing with next gen, and Travis, if you have a good way to explain this, feel free to jump in after. You have really good analogies and metaphors and comparisons. But the short and skinny, the really nice thing with the Sanger sequence, you have one reaction. You're going to get one sequence. So if I amplify both Pied and Clown and I put it onto Sanger, I'm just going to get a bunch of noise because I'll get the sequence from Pied and I'll get the sequence from Clown, but they're on different genes. And so you just get this garbage. But with next gen sequencing, if I amplify Pied and Clown in the same reaction and do next gen, they're all spread out. So they're all separated out. So each individual DNA sequence is its own read. And so I can go into the program and say, hey, I want to see all the Pied reads, and I give it the sequence for Pied, and it will just show me the Pied ones. And I can see if at that one spot, 141, if there's a C or a T or some of both. But then I can say, okay, here's the sequence for Clown. Show me all the Clown reads in this one sample. And it can pull those all out, and I can see if it's one read or the other, it's a one base or the other or both, just like what I did with Pied. And I can do as many as I can get the primers to work together. So in one sample, one reaction on Sanger, you can only do one sequence. Everything has to be the same or you get garbage. Whereas with next gen, I can multiplex as many different things I can get to run together and sequence all of them. And then I can separate it out with the computer and say, all right, just show me Pied. Okay, now just show me Clown. Now just show me Hypo. Now show me Yellow Belly. And I can make all those calls on every single one. So like what you just saw me do for Pied, if you did a full panel right now, I would go through 19 others to make all those calls for you.
Yeah. So
that's awesome.
An analogy, when Ben's talking about amplifying, that's PCR. So he's using basically like a Xerox method. And if you think of it like, the genome's like a book, he's just tearing out a specific page and he's replicating it a whole bunch of times. And that's just for a single sequence. If he's doing multiple different traits, what he's doing first is he's going through the book and he's got a highlighter. And for each of the areas that he's interested in, he highlights it. So for Yellow Belly, he does it yellow. For Pied, he does it green. For Clown, he does it pink. For Enchi, he does it orange. And then he tears that page out and copies it thousands and thousands of times on a color copier. And so when he runs the sequence, he can specifically sort it out, even though he's got millions of copies of each, he can specifically say, look, only show me all of the copies that are yellow, or only show me all of the copies that are pink, or only show me all of the copies that are green. Where like with Sanger, it would just be you're copying the page, you didn't highlight it, and so now it's just like it's trying to cram all of that data together, and you just can't get anything coherent out of it.
See.
It's a completely different ball game.
Yeah.
Yes.
Well, that's cool. Thank you for showing us, Ben. And good analogy, Travis. That's it's starting to crystallize a little bit more for me because I'm still.
These are very bizarre concepts, and they
Yeah.
can take a while to wrap your head around. But
Mhm.
you know, that's doing this years and years and years and years. You start to pick up little tricks and ideas to make it a little less mumbo jumbo and crazy garbage, and a little bit more coherent to understand.
Yeah.
Yep. I
Awesome.
started doing my first sequencing and analysis in 2002. It's been a few years. I did it on cane toads.
Oh, cane toads.
Yeah, I was doing mine about that same time, but I was doing it on bacteria, and
Ah.
I was even further back than Sanger. I was running page gels, so you want painful.
Yeah.
Imagine a gel that's this big,
Yeah.
and each lane is a different piece of DNA that you've chopped up, and you have to look through all the little teeny tiny pieces and find the one that's different between them.
Oh no.
Yeah. And if your gel gets a bubble in it, it gets toasted. If the gel gets too hot and it breaks, it gets toasted.
Oh,
You're
and by the
working with
way,
radiation, and it's not fun.
yeah, and by the way, if you touch the stuff you make the gel with, you get cancer.
Oh yeah, that too. Yeah.
No pressure.
And
Or die.
here's the funny thing. That's the same stuff they use to make water crystals for crickets. So think about that when you're feeding your crickets the water crystals that then you're feeding to your bearded dragons and your crested geckos and all of your things like that. Maybe don't use water crystals. Just throwing that out there.
Today I learned. I'm cool with my colubrid snakes.
Well, for those of you out there, we see some questions rolling in. Definitely feel free. And if you miss something, you want to see some more, I can go back, pull something up. But let us know if you have questions. And Kayla, you're the one that has to kind of be the person that kind of stands in place of people listening, because like I said, Travis and I have been thinking about this for over 20 years.
No, if I think of questions or things that are clarifying questions, I guess I'll throw them out there. But yeah, we do have a couple of general questions in the comments. Like, do you want to take those, or do you want specific?
Yeah. Any kind of questions, totally fine.
Yeah, you got it. So this one's from Connor on YouTube. Do you have an estimate for when monsoon testing will be available?
Yes. So people did react very well when we talked about wanting more monsoon sheds. So I do have enough sheds, and they're sequencing. Yes. Thank you. Thank you for sending sheds in. We had probably 5 or 6 different people that sent in at least one, and some people sent in, 5 or 10. So it made it so we've got plenty. So the first thing will be for that to go to sequencing, and then we'll start the analysis. And in that case, we're sequencing the whole genome. So here we were just looking at part of one gene, those 300 base pairs, because we know where to look already. But in the case of designing a new morph, we have to look at the whole genome, and we compare the ones with monsoon to the ones that don't have monsoon, and look for any differences that are in the monsoons and not in the other. And then we have to PCR around those and test and see if they're truly something that's only in a monsoon and not in something else. And that's that validation process. So for monsoon, you're probably looking at 3 months out still, since we have to finish the sequencing, then analyze, then design, and then validate. So it's a little ways out, but we do have enough sheds. So really, really thankful for that. Thank you, all of you that send sheds in.
Good stuff. Yeah. And like I said, guys, we have said in the past, I guess, it just takes you guys sending in sheds for us to be able to do these. So you done good.
Yes.
It takes lots of people. I mean, it's not that one person sending 50 sheds in is a bad thing, but a lot of times those 50 animals are fairly closely related. So sometimes Ben might pick up a false blip just looking at a real high concentration of those, because since they're all related, they may carry 2 completely different silent mutations that aren't even related to monsoon. but both of those will come up in Ben's thing, and then he's got to chase both of those down, where you've got like 50 different people who send in Monsoon sheds. It's a lot more likely those are going to be unrelated. So those random little silent mutations that pop up, they're going to get weeded out real fast.
Yeah.
Yeah. That's what can take some time, is chasing down those false positives.
Mm.
They look like they could be real, and when you chase them down, they end up to not be. And that's where, I start telling people, oh yeah, this looks good, I'll run 5 and they all match what they should be. I run 20, they all match what they should be. And then I run 100 and they don't all match. Then I know, oh, that's not it, I got to go to another one.
Aw. Okay, we have from Will's Hella Heat. Any updates on Puzzle and Cryptic?
Yes. You can't order it yet, but Cryptic is done. So
Yeah.
That's going to make a lot of people happy.
Mm-hmm.
Yes. We have Dr. Heather Rafi and Dr. Alan Garcia Elfring in Canada to thank. So that was a trade. I traded them Genetic Stripe, Enchi, and Black Pastel so that we could get Cryptic and VPI Xanthic. So just like Cryptic, you can't order it yet, but we do have VPI Xanthic. I have run enough. It's good to go. So we just have to get it all set up with our vendors and to be able to make it available. But yes, VPI Xanthic and Cryptic are good to go. Puzzle, I was told that I will get the data this week. And he said, no, we don't usually swear on this, but I'll give you a direct quote. He said, no bullshit. So hopefully he's going to come through. And so we'll have that data this week, which means probably 2 to 3 weeks, as long as things go well, which Desert Ghost, as an example, they don't always go like we think and could take longer. But if things go well with Puzzle, it could be as fast as 3 weeks out, and we'll have a Puzzle test ready to go.
Yeah. You mentioned Desert Ghost in that one. So TNJ Reptiles asked about that.
Yeah. So since it's baby season, we have tons of samples coming in. We have not put any of the free samples in. So we've definitely run lots of Desert Ghost tests on every run, but they've been paid ones so far because we have so many samples. So we've been running about 700 samples on a run the last 3 runs. They've been gigantic runs. And you have to remember, since we multiplex, some of those are panels, so it's 700, but 100 of them, we're doing 18 to 20 different tests for that one sample. So you do 100 times 20, all of a sudden just those 100, that's a lot of sequence and sequence analysis. So yeah, thankfully there's lots of interest, and it's helping lots of people with their babies they're hatching, which is exactly what we want. But for the free Desert Ghost tests, we have to fit those in where we can, since we have to have a priority for the paid samples. Plus, I think there's still some time for people to send in their free ones. We gave 3 months so that you'd have the time for your animal to shed and be able to send it in time. So it's definitely something, like we said from the very beginning, it's going to take some time. But yeah, the sheds are still coming in, and I would imagine sometime soon things will slow down enough as baby season slows down, and I'll be able to start running some of them. But I don't know for sure when that will be. It could be in a couple weeks. It might take another 6 weeks till samples slow down. I don't know. This is our first year. Mid-October will kind of be our 1 year mark where we've really been doing the morph testing. So we're still learning, how the business is going to run throughout the year. But yeah, I would imagine sometime in the next month or so, we'll start running some of the free ones.
All right. From Tommy Inky Clouds on YouTube, any work on tri-stripe or black Axanthic?
I don't have very many sheds for either, probably 5 or less. So if you know people that have tri-stripes or black Axanthics, tell them to send them in. That's all we need. Once I have 20 to 40, somewhere in there, closer to 40, the happier I am. We'll start working on it.
You got it. From Sloan Morphs at Ben, I handed you a shed at the Pomona show that I paid for through Morph Market. When will I get the results for that one?
Yeah, so we mentioned that in the update at the beginning. But yes, I have your results. Anyone that handed me a shed at Pomona. And so I will try to send those out before I go to sleep tonight so that you guys will have those. It's been a full day. I did have them this morning, and whenever I had 20 minutes, I was going to get them sent out to the few people that handed them to me at the show, but I haven't made it yet. But after this, I will.
All right.
Excuse me.
From Chad Little. Hey Ben, nice beard.
Hey Chad, I'm trying. I'm not up to yours yet, but I'm trying.
Yeah.
Thanks for popping in, Chad. Chad, his table RAL. That was the table Sean was at in Daytona. So
That's right. That's right. Yeah, I think he was in last week's thumbnail, maybe the one before that.
Yep,
Yeah,
he was. Yeah.
Oh, for those of you who didn't catch last week's episode, go look at it. No. It was Friday's post on Facebook, I believe. It had a picture of Ben in the snake discovery video. He looks so different. The beard was kind of there, but it was like really short. Like it's crazy. Seriously, go look at it. You'll get a chuckle.
And instead of wearing a hat, I was wearing a button-up shirt, or I guess a golf shirt, kind of a shirt with a logo on it. So you see me with the hair instead of the hat.
Yeah. And from Andrew Rose, this is the ultimate in DNA nerd talk. Yes. For those of you who didn't catch the beginning of the stream, go check it out. See what like you'll see like the different ways that see the background stuff. That's all the words I have for it. It's pretty cool.
You get to see the mess that Travis and I stare at every day.
Yes. From Gray's Reptiles, we have any update for hurricane testing?
Yes. So hurricane, we still need more sheds. I have had some come in, so that people have heard that, and some have come in, but we still need more hurricane, and same with Trick, and I think there's one other one. Was it Blitz? I can't remember. There's something else that's similar, I believe, but I still need more sheds of those.
Good stuff. So if you know people, like, go ask all your friends who work with hurricane. That genetic diversity is especially important for them to be able to do this.
Yes.
From Jacob Ledbetter of Sybil Serpents, any update on Monarch?
So Monarch is the same situation as Puzzle. So that's the no BS. I'm supposed to have the data this week. So hopefully within a month, we'll have both Puzzle and Monarch good to go. Fingers
crossed.
I'm interested to hear about that one.
Yes, that will be interesting. So which part of the drama are you most interested in?
I'm interested to find out if it's just another allele of the Ultramel complex. I would like to see that information.
Do you have any guesses?
I don't know. There are some days where I've seen some monarchs that look just like really, really, really dark, ultramels or something. Or I've seen ultramels that just look like I could have mistaken them for a monarch. And there are other days where it's like, Lavender and Candino look a lot alike and they're completely different. So don't necessarily stick something in the same box just because it looks the same.
Yeah.
And Patrick McKnight, I know, was on earlier in the stream. I don't know if he's still on, but Chimera Reptiles, he's got something that popped out of his Ultramel line that's freaking crazy. I don't know how much.
Does he have one of those weird, God, what are they calling it now? Mimic or something?
I don't know if he has something that other people. I know that, and I won't give anything away. I'll let him share. Like I said, I don't know if he's shared it publicly, but it looks
it's something weird,
different from other Ultramels, and it's cool. It's very cool.
Nice.
Yeah.
This is from, I think you pronounced that, Denise Reptiles. For a newbie, can you explain what the het means in terms of genetics?
Yeah, so if something's heterozygous, what happens is when a sperm and an egg meet, you get two sets of chromosomes. So in people, we have 23 chromosomes that came from our mom, 23 chromosomes that came from our dad. And so for each gene, we have two different copies. We have one that came from our mom, one that came from our dad. And so if there's a mutation, it could be that just your mom had that mutation or just your dad had it. And so I could end up getting, it could be for eye color, it could be for some specific protein, you know, in my toenails or whatever, any of these different genes. All the heterozygous means is that I got one type of a copy from my dad, but I got a different one from my mom, or vice versa. So heterozygous means different. So you have two different copies. In the case that we showed earlier with the piebald results, visual pied had all T's, a wild type or a normal had all C's, and then the heterozygous animals had T's and C's, about half of each. You have another good way to explain that, Travis?
Sort of. So the way I like to think of it is, imagine you've got, like you said, you've got the 23. Imagine they're card decks. So if you get from mom and dad the ace of hearts from both, you're homozygous. But if you get the ace of diamonds from dad, the ace of hearts from mom, now you're heterozygous. So it's the same gene, it's just whether or not it's the same suit in the deck of cards that you get. In the hobby, it's, I don't want to say exclusively, but almost exclusively used to refer to recessive genes, where the het is carrying a gene that's basically silent because the wild type gene covers it up. But the reality is that anytime you have two different alleles, it's a het. So when you look at like a Mojave ball python, that's got two different genes or two different alleles of that gene there. So that is a het. It's a het for blue-eyed leucistic. We just don't call it that because since it's visual, we just call it, you know, the visual Mojave instead of a het blue eye.
Yeah, that's a good analogy. I've got a couple comments praising that down in the down in the comment section. So I like that.
I'm glad they like it.
Yeah.
Yes. That's what we love, having Travis come on.
Yes, we do. And thanks for asking that question too. It's awesome to see new folks getting in on this. And sometimes we throw around the verbiage without, you know, maybe explaining it. So good stuff. And is that a cotton? I think that's is that a copperhead or a cottonhead in their profile picture?
That looks like a very dark copperhead.
Yeah, it's cool. I kissed her on for sure, but yeah. Anyway, so we have, skip one. From Prophecy Exotics, any update on acid and confusion being the same mutation?
So I definitely have run enough confusion samples. I do have a pretty good location, and like we talked about earlier, kind of going through any false positives to make sure I have the correct spot, the correct, have a mutation that will help us have a test that will be reliable over 99%. What I don't have, should have got water. My throat's dry.
It's all good.
What I don't have is very many acid samples that I've run yet. So I don't know for sure about that, but we'll be able to figure it out.
I have this whole box of sheds that I really need to get to.
Yeah. Oh, me too. I have a bin.
A bunch of those have got acid in them, and I know a guy. I'll stick a fork in him and get him to send me some acid too.
Awesome.
Yeah. Oh, and Denise Reptiles answered, it is a copperhead. That's the darkest copperhead I've ever seen. That's awesome.
That is pretty.
I think it may be in blue, and that's why it's so dark.
You know, that would explain. Yeah. Awesome. Cool. From Terry Pruitt, any insight on Enhancer coming, or did DG take all my thunder? Sad face.
So far with Enhancer, I definitely see both DGA and DGB in enhancers when people, once again, I have to go off what the breeders and keepers tell me when they send the sheds in, but being reported as enhancer, I still see that. But it would not surprise me at all if we find out that, you know, DGC or D, as we find, you know, more genes that play a role in causing an animal to be a visual desert ghost, we might find something that's more commonly in enhancer or less commonly in enhancer. But definitely DGA and DGB, I see that in enhancer samples as well.
All right. From Mike Calderon, lots of people say Puzzle and Cryptic look very much alike. Can you confirm that they're different morphs?
Yeah, so as everything that I know as of right now, they definitely are different. I will know this week as long as the data really comes through. No BS, hopefully is true.
Well,
I'll be able
if memory serves me, I believe I have seen puzzle clowns out there. So that would very much indicate that they are not the same, because you couldn't have a puzzle clown, because that would be 4 copies of the same gene in one animal.
Yep.
So if puzzle clown exists, then and cryptic we know is an allele of clown, then puzzle and cryptic are not allelic.
Yeah.
Very
Yep.
cool.
So yeah, I haven't seen any evidence at all. That I can see where, you know, you kind of get a wide eye stripe and you get kind of that funky pattern. But yeah, everything that I've seen genetically, they're on different chromosomes or very far apart on the same chromosome and not linked at all.
Yeah.
All right. From Angel Garcia. So I saw new morphs such as gorilla and tornado. How do you go about classifying or declassifying these morphs?
We depend 100% on you all. You all have to send sheds into us, and there's nothing, absolutely zero I can do until I have sheds that I can sequence and compare and look for differences. So
And with a lot of these new morphs, that's going to fall back on the it's usually one person who's found it and come up with it. So any of the sheds that are coming in are all going to be very related to one another.
Ooh, yeah.
And that could very much give the false positive type of thing that would throw Ben off. So with a lot of things like that, if you have suspicions that it's related to something else, maybe try breeding it to that something else. Like if you get something that looks kind of like a black pastel, maybe breed it to a black pastel and see if you get, a super form or one of those compound heterozygotes, like an onyx or a slick or something like that. And that would give you a better idea of if it's in the same ballpark, because then when you send that shed into Ben, you can say, I've got what I think is a new morph, and I bred it to a black pastel, and it looks like I got a super. So at least then when he sees hits coming up, if they're in the same spot or close proximity to the black pastel spot, he'll know that he's looking in the right direction.
And that's a gigantic help. That narrows it down from 1.5 billion base pairs, usually down to, less than 5 million. So that's very helpful. It's still a lot and it can still be a lot of work, but that narrows out a lot of potential false positives.
All right. From Noah Gardner, have you thought about using scripting to automate any part of the process, like Python, R, MATLAB, etc.?
So I actually showed Kayla and Travis what I do with Python. I do have a Python script that helps me. So what I showed today is a little bit, more complex than what I do on every sample, but then you have to keep in mind that I'm doing like 700 samples. So it's still tons of genetic information I'm looking at. But yeah, you're definitely right. I use Python scripts to help me do some of the analysis for sure. I wouldn't be able to make it through all of those samples without having some scripts helping me out.
Yeah. We almost tried to make a pun on today's like live stream title, like something with Python code.
Decoding Pythons with Python.
Yeah, exactly. But we didn't know how niche it would be. Like, I don't know. Sound off in the comments if you would have gotten the joke if we read it.
I told her it was probably too nerdy.
Yeah. Let's see.
Noah would have gotten it.
Oh, Noah would have gotten it?
Yep. Yeah. Noah, the ant asked that question. Yeah, he would have gotten it.
Yeah, that's true. So that's at least 4 of us. 3 in the stream. From Insane Clown Pythons, are you working on the NR Mandarin test yet?
That one I need more sheds still, and I have had some people start sending in some, but I definitely need more. There were multiple people at Pomona that said they have several animals that we're going to start sending them in. I haven't seen them yet, but if you know people, if you have them yourself, Mike, or know people that do, get on their backs, make them get those in. That would be a good test. I think that would be really helpful, and especially if you do along with hypo, if you test both Mandarin and hypo at the same time, I think that'll help kind of get things figured out for people, some of the questions they have around that morph.
There you go. So tell your friends. From Mike again, if you're running Python script, you should get some programmers to look at your script to see if they can make it more efficient.
Yeah, and I very well may do that, because it's getting to a point where I've got a lot to get through.
Yeah.
These last ones where we have so many samples, it's taken me 2 or 3 days to get through everything, focusing pretty hard for several hours. So I'm definitely a biologist who knows a little bit of bioinformatics and scripting and computer science language. Very little. I've done some Python and some Perl, and that's like it. And so I usually just take what somebody is already made that I know is similar to what I want to do, and I go in and just make some little changes, and that's the level that I understand it. so yeah, I do. When the samples slow down, that's something through the winter and spring, when we don't have as many samples coming. I need to get that updated. And then Mike, if you have some suggestions for me, and Travis as well, I haven't asked Travis, but either if you yourself or someone that might be a good person for me to talk to, I could use some help with that for sure.
Yeah.
I can talk to some of my bioinformatics guys at work and see if they could reach out to you. I'm sure a couple of them would be willing to.
Yeah, that would be good. I'm in a spot now where things have grown a lot. So it's a different ballgame than what we were doing a year ago.
Yeah. Noah said, my coworker said, I don't know how you can work on Python all day and then go home to more Python.
Or use Python to analyze Python data.
Yes. Hey, Tracy Sweet or Tracy Sweat, whichever. I'll say both. I totally would have gotten it. My oldest son is learning to program basic games in Python this year. So
that's awesome.
There's some more people that would have gotten it.
I think anywhere where they're teaching kids to do coding, I think that's awesome. I'm the oldest of 9 kids, and I tell every single one of my siblings when they're going into college, whatever they're interested in, also take some computer science classes.
Mm-hmm. Let's see. And I think we got this question earlier, but from Willie B Exotics, I guess they just joined the stream. Did the DG free sheds get done? And if so, Dr. Ben already did the results.
Yeah. So on the free ones, this hit us right as we're in the busiest season with all the babies.
Baby season.
Baby season. So we definitely have to fit the free ones in on runs with paid samples, and the paid ones definitely will get higher priority. And people are still sending in the free sheds because they had 3 months to send them in. So it's something that will take time. But as we start to get those run, we'll definitely have some updates on here as we start to get results. I also hope, we didn't say this earlier, but another thing is the no BS data I'm supposed to get this week that we talked about earlier does include a bunch more Desert Ghost whole genome sequencing. And so I'm hopeful that by the time we're running the free ones, I have a DGC and or DGD, So that way, when I go through and run these hundreds, I think we're at 300 or 400 samples that people have sent in for the free test. Excuse me. Mm-hmm. It'll be much better if I start running that once we can do DGC and or however far we can get. C might be as far as we can get. I'm pretty confident we can at least get one more gene that's a big contributor to that visual expression of Desert Ghost. So hopefully the wait is going to be extra worth it, because you'll get even more data that will help you know even better how likely your animals are to produce babies that are visually Desert Ghosts. But yeah, it's coming. A project like this, even if you have a chunk of money to run it, it still takes time. I'm sure Travis has been in the same boat. There's many projects I've been a part of that take 3 to 5 years. Yeah. Anything you get done in less than a year is pretty dang fast.
Oh yeah, that's really speedy.
Yeah. So this kind of stuff takes time. And even once we get the data and start giving people results, then what we want to do is figure out a way to present it visually so that those of you that are interested, either that submitted samples or you just have have an interest in analyzing, we want to be able to put results with a picture so people can look at different genotypes and the picture along with it. And you can kind of go through and see for yourself, what potential differences are, maybe how you get more of a yellow or more of a axanthic look. And so it will take quite a bit of work to visually present all that data. And I might have to go get a glass of water. I've never had my throat get this dry during this.
Oh no, yeah, no, that's fine.
But yeah, it's going to take some time, but I think it's going to be really, really interesting and really cool. And you all will have a chance to check it out.
Yeah. Actually, if you want to go grab that, I think I can handle the next
All right,
two.
I'll be back.
Yeah, go ahead. Okay. From Constricted Reptiles, Dale Richard of Constricted Reptiles, I have about 20 something acid sheds for you. So awesome. I think between you and Travis, I think we'll be pretty good on acid.
Yeah,
pretty sure.
Dale's a good point. Yeah, he's one of the people that I was going to hit up and ask him to send sheds in. So
perfect. All right. He's the guy you're going to be poking at then.
Yeah,
awesome.
him and Josh Jensen. I mean, and Josh is the guy who originated the acid. So I'm sure that Josh has more than a few kicking around.
He might have one or two.
Yeah.
Awesome. And from Silly Serpents, if you have my tri-stripe POSHET DG test done, tell me live. We would love to do that, to do a live test. Let's see. I'm not sure how soon we'll be able to do DG tests, like live.
If it's a paid one, then it will get done. I don't know.
Okay.
If it's a POSHET, then it should be a paid one. So I'll try to keep an eye out for it. Yeah. We've got, dozens to hundreds of those per run. So it'd just be if I get lucky and catch yours. But if you message probably to either Instagram or Facebook message your order number so we know which one to watch for. If we happen to catch it and can do that for you, we'd be happy to.
There you go. Let's see. Insane Clown Pythons wants to know if they're getting their results today.
Yes. Coming soon.
Coming soon. Yes. All right. I think that's all the questions we had. And we are closing in on almost an hour here. So, all right. Ben, Travis, do y'all have any last thoughts or anything like that? Last thoughts. That sounds morbid. Last minute thoughts.
Closing thoughts.
Closing thoughts. Yeah.
I mean, no, I think it's, it's really great to see the level of engagement that this is bringing out to the community. I mentioned this to you guys before. Like, I remember on the Bush League days when I brought this up, and everybody's like, "Why would we want to do that?" And feeling kind of defeated at the idea that everybody thought that genetic testing was just dumb. So seeing that it has become this really big popular thing is really kind of cool. And I enjoy the fact that you guys like to have me on here and help explain things like this, because it's a great way to bring the collective together and get everybody on the same page with stuff.
Yes.
Absolutely. Oh, another question, also from Insane Clown Pythons. You guys coming to Arlington? I am.
Yep. If you say by you guys, you mean Kayla. For Travis and I, unfortunately, no.
Yes.
It's a little bit too far for me.
It's fair. I'm flying it down there because Ben can't make it and Sean can't make it. So I'm gonna roll down there from Virginia. We'll have merch and shirts, and I will answer whatever questions I can. We might have the other part-time worker who's been helping prep samples. Allie, yes. We might have Allie there too. Not sure yet, but I definitely will be there. So,
well, I can make myself available. You can hit me up by messenger during the day, and I can try to get answers to you as fast as I can. But I can't guarantee I'll be in front of my computer all day either.
Fair.
I will definitely be thinking of you too, and if I can answer, I certainly will be happy to.
Yeah, sounds good.
Sean, on the other hand, he won't get back to you. He's going to be out of cell range.
Oh yes, he's going to be out elk hunting, I think, something like that. So yeah.
Yep, he'll
All
be
right.
off to the races.
Out on Sean adventures. All right. Well, let's see. Tracy Sweat says we need to come to the Northeast Expo sometime.
Where's
I don't
that
know when
one?
that one
Yeah,
is.
which one is that?
Yeah. Northeast Expo.
Is it New England Expo?
New England. Yeah,
that
that
what
would
it
make
is?
sense. England Reptile Expo.
Yeah.
That's in September 10th, 2023. That's my birthday. Well, I won't be there. I'm going to be at somebody else's wedding, actually. But
That's one we'll have to keep an eye out for. Is it in Maryland or where is that one?
That one.
I imagine that's further north.
Says White Plains.
Oh, White Plains.
Oh, White Plains. Okay.
Yeah, I've heard that show.
White Plains.
Nice big show. I would like to come to that one. I've gone to the Hamburg one. That's the furthest in that direction that I've gone. I haven't made it up to New York.
Yeah, okay. I don't know that I'll be up to traveling up to White Plains in another 12 days.
Yeah, maybe. And from Bruiser Captive Bread, have you happened to work on any fire yet?
Yes, fire is another one like Confusion, where I have a very good location and just kind of weeding out false positives. But yeah, that one we should have fairly soon.
All right. Armiana wants to know, did the secret complex we mentioned a couple of weeks ago ever get announced outside of the show?
So that one you'll have to watch Eastern Michigan University. I don't know for sure when she's going to announce that, but that's a complex that they worked on and have figured out. So
yeah. All right. I remember, I think Ben told me, but I've already forgotten. I'm sorry, guys.
That's so secret. Even if you find out, if it's too early, it leaves.
It just leaves your brain.
Yeah.
It's gone. Erased.
But yeah, I would imagine she'll announce that within the next month or two.
That'd be awesome. All right. Closing in on an hour here. I guess we'll call that a wrap.
Yes, thank you so much for watching. Like Travis said earlier, it's something that we've nerded out on for a long, long time, and it's really been a lot of fun. I went on a reticulated python. Man, my throat tonight.
Yeah, that's okay.
I went on a reticulated python podcast a couple nights ago that will get released this week, and just other species and other types of questions, localities, and how long an animal is going to get. A lot of different things that I think we'll be able to come up with tests for eventually. And
yeah,
that would very be cool. Fun, very cool. Seeing people get excited and learn, and be patient with us as we grow as fast as we can. And it's been awesome, and really appreciate it.
All right, awesome. Well, thanks again for everybody who showed up to the stream tonight. We kind of covered a broad range of topics, and it's always a good time answering questions and engaging with you guys. So thanks again, and I guess we'll call that a wrap. Outro in 3, 2, 1.